Hi guys! Today we're talking about chromosomes. So before we get in too deep, let's remember there are two major types of cells, prokaryotes and eukaryotes. Prokaryotes are typically bacteria. They're single-celled. They don't have a lot of organelles. They have some, but no nucleus, right? Eukaryotes are different. You are a eukaryote. Eukaryotes can be single-celled or multicellular. They have complex organelles like the endomembrane system, nucleus, things like that. Now, both prokaryotes and eukaryotes store condensed DNA in the form of chromosomes, but they do so differently, and we're going to talk about those differences today. So, like I said, within both prokaryotes and eukaryotes, DNA is condensed and stored in the form of chromosomes. Here we have a prokaryotic cell, a bacteria, and we have a big long loop of DNA. Now you can tell it's DNA because it's double-stranded, and here you have your complementary base pairs. That's important. So, here is your prokaryotic DNA, one big, long, large loop, and you can see it just kind of like hangs out inside the bacteria. However, a eukaryotic cell, like your cells, you have a nucleus, right? And your DNA is stored in the nucleus. Now, most of the time when your cells are not in either mitosis or meiosis, DNA is stored as a long, loose thread of chromatin. But we're going to be talking about the coiled and stored packaging of DNA when it's getting ready to go through mitosis. You can think of chromosomes as like moving boxes, right? You put all your stuff in boxes, nice and organized and labeled so you know what room they go to. Well, your DNA coils up into a compact storage container just like that, and that's what a chromosome is. You can see that the DNA is tightly wound like a slinky. And the DNA molecule itself is wound around these molecules called histones. And we'll talk more about that today. So, big differences. In the prokaryotic cell, DNA is loose in a long singular loop. In eukaryotic cells, you have this tightly coiled, slinky-like shape with DNA wound around proteins called histones. And don't forget, the nucleus. And that's important, because otherwise, if we didn't have a nucleus, both transcription and translation could all happen out here. But because we have this membrane that protects our genetic information, we have to do transcription on the inside and translation on the outside. Eukaryotes got to be complicated. They got to make everything complicated. Okay, so let's get into our understandings. Understanding number 1, prokaryotes have one chromosome consisting of a circular DNA molecule. Understanding number 2, some prokaryotes also have plasmids, but eukaryotes do not. So here's a better diagram of a prokaryote. So prokaryotes have one chromosome, singular chromosome, consisting of a single strand of circular DNA. Now that makes sense because they're simple organisms. They're single-celled. Their genome, or the total amount of genetic material that they have, isn't going to be very big. They're not super complex. They're pretty simple. So they only have one chromosome. We say it's naked, meaning there's no nuclear envelope, and it's located in the nucleoid region. And the nucleoid region is where ribosomes are produced. So here we have our prokaryote, we have the capsule, we have the pili, which are the little hairs that stick out, we have the inner cell membrane, right? Here's the inside, cytoplasm. This is our chromosome, and it's in the nucleoid region. Right? Here's our single looped chromosome. It's all coiled. No histones. It's just like a ball of loose string. And then here we have this other little thing. It's called a plasmid. And it's just a little circular loop of extra DNA. Eukaryotes do not have these. So plasmids are important. They can be responsible for traits like antibiotic resistance, and they can be transferred through a process called conjugation. So most eukaryotes, pretty much all multicellular eukaryotes, we use sexual reproduction, right? And that helps us mix up our genetic material. That's why you have genetic material from 4 of your grandparents, right? Well, prokaryotes can't do that. They don't have sexual reproduction. They use binary fission. So they basically just make copies of themselves. Well, that kind of limits evolution because unless there's a spontaneous mutation, you have no recombination of genetic material to garner new traits. So instead, bacteria came up with this like funky little way to trade bacteria without sexual reproduction. They basically just like trade DNA. It's crazy. That would be like if I had a certain trait, say I had a trait for like, I don't know, a superpower. Like I could shoot laser from my eyes. That would be like, hey, I'm going to give you my trait of laser eyes. And I just like touch the person next to me and like poof, they have laser eyes. Now, it sounds kind of crazy, but you got to think if there's no other way for changes to occur in your population, evolution's going to happen really, really slowly. So this capacity to like trade genes whenever you feel like it does a lot to enhance evolution in prokaryotes. They can do it extremely quickly. So here's our bacteria. Here's our one chromosome, right? And then here are these extra little loops of plasmids. And they can just like trade these back and forth, kind of like trading cards. You'll hear this called lateral gene transfer. Now, there are other ways besides conjugation that bacteria can trade genetic information, and it just basically includes the use of a vector. But for intents and purposes, you can kind of just see what's happening here. This is like the easiest way to understand it is through lateral gene transfer. What you're looking at is a GIF that's going to show you have a bacteria that has a plasmid for antibiotic resistance. So say this is like a staph bacteria, right? Now, this bacteria can give this bacteria that trait for antibiotic resistance just by being like, hey, guess what? I have an extra copy. Here you go. Now, both of these cells have the plasmid for antibiotic resistance. They duplicate through DNA replication, and then they can give it to other bacteria. This is why it's so important to use antibiotics sparingly, because the process of sharing antibiotic resistance as a trait in bacteria can spread extremely quickly. This is how you get MRSA infections, so resistant staph infections, or TB resistant tuberculosis that can't be treated with antibiotics, or syphilis that can't be treated. It's pretty devastating because once you don't have a medicine to treat it, you're essentially, stuck with whatever disease, and you have to hope it doesn't kill you. Okay, understanding number 3. Eukaryote chromosomes are linear DNA molecules associated with histone proteins. So we're done with prokaryotes now. Now we're going to look at eukaryotes. Histones are proteins found in eukaryotic cells that bind DNA into tight coils. Linear double-stranded DNA molecules wrap around these proteins and condense. This allows great lengths of DNA to be stored in a very compact space, and we call that supercoiling. So here's our DNA. We wrap that DNA around these proteins, they're called histones, and then coil them all up, and you have all, or not all, but you have a significant portion of DNA condensed into a chromosome. Again, prokaryotes do not have histones. They don't do this. Now, we only do this when we're getting ready for mitosis or meiosis. DNA, when it's in the chromosome form, is difficult. It's much more difficult to access the DNA, so you don't have as much transcription going on when the DNA is wrapped up in these chromosomes. So we don't want to keep them wrapped up like this forever, right? This is only when we're about to go through cell reproduction. So here's another diagram. You can see we have this scaffolding backbone. We have the DNA itself, which is this little black thread that's surrounded by nucleosomes. And then you basically keep coiling them around each other, and you get this kind of slinky loop. Now, you're not going to have to draw this, but you do have to understand that you've got multiple levels of coiling, right? You've got DNA coiling around the histones, and then you've got the coiling of the entire thread altogether. Understanding number 4. In a eukaryote species, there are different chromosomes that carry different genes. Understanding number 5. Homologous chromosomes carry the same sequence of genes, but not necessarily the same alleles of those genes. So, if you're a eukaryote, you have 2 or more sets of chromosomes. Now, humans have 2. Plants can sometimes have like 6, 7, 8, because plants are awesome and they're better than us. But let's go back to like humans for a second. You have 2 sets of chromosomes. You have one set from mom and one set from dad, right? So this is chromosome number 1, and you have 2 copies. One from mom and one from dad. I can spell. D-A-D, right? This is chromosome number 3. You have one from mom and one from dad. Now, chromosomes, like I said, are moving boxes, and the same genes are located in box 1 from dad as box 1 from mom. And that's important. When we talk about crossing over in meiosis, you need to make sure that you're crossing over genes at the same place. So you can kind of think of chromosomes as like a neighborhood city block with a bunch of houses on it. And the addresses for the houses on your dad's block have to match the addresses on your mom's block. So if the gene for eye color is right here on dad's chromosome, the gene for eye color is going to be right here on mom's chromosome. They have to be in the same location. Eukaryotic chromosomes exist in sets called homologous chromosomes. Each member of a pair contain the same genes in the same loci. Loci means location. Loci is plural. Locus is singular. Let me see. Locus equals singular. Loci is plural. So here we have sets of homologous chromosomes. This is just like the image I showed you before. It's just a little neater, right? So here we have chromosome number 1, chromosome number 2, chromosome number 3, 4, 5, right? And you have one copy from each parent. So a copy from mom and a copy from dad. That means if this is the gene for ear shape, this is the gene for ear shape on dad. They have to be in the same location. Homologous regions are indicated by the shaded and unshaded bands. So you can see, like, here's an unshaded band, that means they're the same, right? And here it's the same. If you were in my class freshman year, you may remember the pop bead lab where we had the different colored beads and we made the chromosomes, and you were in-laws and you were shuffling genes up for your grandbabies. Well, I think we're going to do that again next week. Okay. Understanding number 6. Diploid nuclei, represented by 2N, have pairs of homologous chromosomes. Understanding number 7. Haploid nuclei, represented by just a single N, have one chromosome of each pair. Most cells have complete pairs of homologous chromosomes in their nuclei. This is referred to as being diploid, right? This is when most cells in your body, so pretty much all cells in your body except your gametes, right? So all your cells except for your sex cells have 2 sets of chromosomes, right? One from mom and one from dad, right? Your gametes are haploid. So haploid cells contain one chromosome from each pair in their nuclei. Gametes are haploid and must fuse in order to form a diploid cell. So when we say gametes, we mean eggs and sperm. Eggs and sperm. Every other cell in your body, every other cell is diploid. Brain cells, tongue cells, butt cells, they're all diploid, right? Haploid cells are formed from diploid cells during meiosis, which we'll talk about in topic 3.3. There's meiosis 1 and meiosis 2. We're going to talk all about that soon. We'll also talk about mitosis soon because we skipped it last semester. Understanding number 8. The number of chromosomes is a characteristic feature of members of a species. Application number 3. Comparison of diploid chromosome numbers amongst species. So each species has a characteristic number of chromosomes which are shared by all members of that species. Examples of chromosome counts are shown here. So here we have a worm, some rice, and we have a chimp. So here is our rice. They have 24 chromosomes. Homo sapiens, us, we have 46 chromosomes. Chimps have 48 chromosomes. Yes, that's correct. They have 2 more than we do. Most of all, domesticated dogs. They have 78 chromosomes. So, the number of chromosomes that you have does not necessarily indicate complexity. Now, when we're comparing prokaryotes and eukaryotes, it does, because prokaryotes only have 1 chromosome. Eukaryotes have more than 1 chromosome. Even if they only have 2, like that worm, they still have more than 1. But chromosome does not necessarily indicate intelligence. Although chimps are pretty smart, so are dogs. All right, I'm going to pause this so the video doesn't get too big, and I'll start re-recording in a second.